Hi, this is the video abstract for the paper Violence in Latin American Preadolescents, a study of social brain function and cortisol levels. I am Buchweitz, the first author, and this is to appear in a special issue of Developmental Science. The goal of the paper was to investigate violence and its association with brain function and hair cortisol concentration in Latin American preadolescents. Latin America is home to less than 10% of the world's population, but it is characteristically a very violent region. Youths and adolescents in this region are affected disproportionately by violence. For example, in Brazil, 80% of youths in school report that they think their schools are a violent environment, and about 70% report being victims of psychological or physical violence at school. So how did we go about carrying out the experiment? So we engaged adolescents in schools in our hometown, and they answered first at school a questionnaire on victimization, which is called the JVQ, Juvenile Victimization Questionnaire. It gives us scores on several types of violence and victimization, which include both physical and psychological violence. After the kids performed this part of the task, some of them were invited to the Brain Institute, where we collected 3 centimeters of hair samples to measure cortisol concentration, and where they performed a task in the MRI. The task is called the EYES test. The EYES task is a social cognition task. Basically, it involves 2 conditions. In one, participants are shown pairs of eyes, and they have to decide the mental state in which the person is in. In the other condition, they're shown the same pairs of eyes, but they have to decide if it is a man or a woman. So what does the literature show? Stress can result from violence and can impact body and mind in different stages of life as an effect of recency and chronicity. Adolescence, specifically, is a period of heightened susceptibility to the environment. To illustrate, this is a security video from one of the schools in our city. As you see, the kids are playing during break and then they run. This is not because break is over. This is because there was a shooting nearby. So the analyses we carried out were correlational among exposure to violence, brain function, brain connectivity indices, and cortisol levels. So the first result showed a negative correlation among the exposure to violence and the activation of 2 regions. To illustrate, this is one of the regions in the right superior temporal sulcus. And basically what we're showing is the activation of different participants in different color codes. So the participants who were exposed to less violence are in red, and then it changes to shades of green and blue. Our other result showed that specifically in the mental state condition, the activation of both the left and the right side of the brain's amygdala and right fusiform gyrus correlated negatively with exposure to violence. So a z-score for the correlation of the time series of activation among these 2 regions correlated negatively with exposure to violence in the mental state condition only. Finally, our biological measure of stress, hair cortisol concentration, correlated positively and significantly with our adolescents' reports of exposure to violence, giving us an association among a biological measure and what these kids are telling us about their day-to-day lives. In sum, our study shows these associations among exposure to violence and biological and neurobiological function and markers. We cannot speak as to whether these markers are early signals of psychopathologies, but the literature does show that a typical amygdala and right hemisphere brain function can be associated with later risk for psychopathology, mood disorders, and behavior problems. This is the first time that a Latin American population of teenagers has been investigated in terms of the effects of brain function and violence.